have a 2-carbon compound. This compound binds citrate synthase, which is the first enzyme in the citric acid cycle. Okay. So, the other thing to mention is that this pyruvate dehydrogenase complex has 3 different subunits, E1, E2, and E3. But it unfortunately doesn't just have, or fortunately, depending how you look at it, it doesn't have just one copy of each of those. It has like 30 or 40 or 50 copies of each of those. So this thing is just this is a cartoon of it. And so E1, here are all the E1s of these little purple balls. So they're all part of one enzyme. Right? So this enzyme is 7,800,000 molecular weight. Okay. It's made up of 3 different kinds of subunits, E1s, E2s, and E3s. You can see the names of those 3 enzymes. 2 of them are named after lipoic acid, the cofactor we haven't talked about yet. Right? And so if you look at this cartoon and kind of translate, you can see there's some E3s in green. This pink stuff is hiding in there somewhere. The blue ones. Right? So we have a situation here where we're going to have a multi-step reaction that all occurs on the surface of this enzyme. But because there's so many different places on the enzyme where it can happen, that reaction can be happening in a lot of different places at once. So you can have a lot of throughput. So rather than having 30 different individual enzymes that do this, it's got them all together in a particle. So that has some metabolic importance, presumably. Although exactly what it is not really that well understood. Why shouldn't it be that we, it's complicated enough, isn't it, to make it a heterotrimer and say E1, E2, E3, okay, that's what you need for one reaction, then that's what you should have. But instead we have E1, E2, E3, times 30 or something, all in one big particle. This isn't the only enzyme, by the way, in the TCA cycle that's organized this way. There's another one, and I'll point that out later. Step 4 or 5 is also organized this way. The only difference between the 2 is what the incoming substrate is. In this case, the incoming substrate is pyruvate. The other case, it's a different metabolite in that cycle. Okay, so we have something going on here that's called channeling. And I'm not sure if I have a slide on this, maybe the last, yeah. So the channeling in this case is going to refer to 3 steps in the pyruvate dehydrogenase complex reaction. It's really actually 5 steps in this reaction, but the substrate is going to get moved across the surface of E1, E2, E3 on the surface of this particle. And so what we're used to talking about are enzyme reactions that are not channeled. Right? Like when we talk about glycolysis, the way we've talked about it is to say, okay, hexokinase releases glucose 6-phosphate, and phosphohexosubstitutase binds it, and it releases fructose 6-phosphate, and blah, blah, on and on. And they're all just one enzyme at a time, releasing, binding and releasing, right? And so that would be what's diagrammed here. Enzyme number 1 binds substrate, converts to product, releases product. Enzyme number 2 binds product, etc. So no channeling. Okay. It turns out, by the way, and I didn't mention it before, that there's increasing evidence that there is, in fact, some channeling in they're very weak, then they just break up in your test tube. And now you've lost the capacity to study it in association because it was really weak and it only held together in the cell. Because your pH 7 tris buffer reaction is not anything like what's going on in the cell in many respects. Right? A channeled complex would be if you have, say, E1 and E2 bound in a complex, actually bound together, so that when the product forms on one, instead of dissociating and rebinding, wouldn't it be more efficient if the product could just kind of move over the surface? Or maybe it could shoot through a tunnel, What if the enzyme had a tunnel in it, so that the product of reaction 1 shot through a tunnel and emerged in another active site and got reacted again there, got facilitated for another reaction there. So there's actually plenty of enzymes that do that. And so when we're talking about this channeling in the pyruvate dehydrogenase complex, we're going to be talking about different steps on the surface of this enzyme that channel the intermediate through the enzyme. Right? We'll still be talking about only one reaction here, one overall reaction, and we'll come back to this next time. But the overall reaction on this enzyme, E1, E2, E3, is going to be to move pyruvate in various aspects that get split up pyruvate through this enzyme, and it stays on the enzyme the whole time. Right? So in that sense, we could say it's channeled. So we'll come back to this and look at lipoic acid and figure out how these reactions work next.